Right. Okay, that today what we are going to do about ovulation, right? Ovulation is not only common to the humans, and also it is noticed in other, animals who, like dogs, who deliver babies. So this is a very normal process, right? So that in this section we are discussing ovulation and fertilization and outcome until cleavage. So these terms we need to understand: ovulation, cleavage, and describe the process of cleavage and blastocyst process of blastocyst formation. Name the site of normal implantation and list the sites of abnormal implantation and its consequences. List the types and causes of twin births and explain multiple pregnancies. Define embryonic stem cells and their possible application in clinical practice. So this is in your very first year. So you are learning these things, right? So next one, that means the very first few days in your medical college. So these are the things what you normally learn under anatomy. So, but under anatomy, so another subsection, embryology, right? So this is the ovary. So that is the cross, is a diagram of the ovary actually, right? So ovary, the size of the ovary. So it is a 3D. So size of the human ovary, right? Is a very small, right? Not a big structure, very big structure. If you so you can get the idea, so that you know if you see the ultrasound scan or other scans, so normally they that means if you scan the ovaries only or abdomen for some reason, if to the ultrasound scan is the easiest way because it's no because there's no radiation exposure. So normally it is 3.5 centimeter, 3.5, 3 or 4 into 2 into 1. So that means the volume is very small. It's about 6, right? 6 centimeters. That means 3 into 5. So 3 into 2. So 3 into 2 is 6. No? So that volume is about 6 centimeters. So technically, it is that means if you think about the volume in millimeters, so 3 to 6 millimeter in volume. That means just like a teaspoon. The volume, that means normal tablespoon volume of the tablespoon is what, 10 millimeters, right? So the volume of the ovary, it is 3 into or 3.5 into 2 into 1. That means about 6 centimeter cube. 6 centimeter cube. So what does it mean? This is very, small structure. So ovary is a very small structure and very delicate, right? So if I write to the size here, so 3.5, right? 3.5, 2 into 1. 2 into 1. Volume is about 6 milliliters. So what does it mean? Extremely small, right? So the structure of the ovary you need to know. So you get two regions, medulla and cortex, or cortical region and medullary region, right? So now here the structures you need to know. So arteries and veins, germinal epithelium, tunica, albuginea, then primordial follicle, pre-ovulatory follicle, growing follicles, corpus albicans, and corpus luteum, right? So it is unlikely, like O levels or A levels, giving them to asking you to draw it and label, unlikely, but possibility also. Therefore, it's better to know. So you will be shown a slide. That means a prepared, that means a fixed slides. So they have, in anatomy department, there's a section embryology. So, or sometimes if it's a small university, the embryology also do it the same department. So you can, so they prepare these slides, or in But otherwise, they import from US, UK, prepared slides. So they colored fixed actual one, so you can see the slides. Or right, or sometimes they have preserve it is in bottle, so you can see those things structures, right? So now here the surface of the ovary is covered by a layer of cuboidal cells. So cuboidal cells are epithelial, that means a cuboidal cells. So those are just so no, so cuboidal. So cuboidal means something like that, I guess, no? Cuboidal, 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 cube. It's just like a cuboidal, right? So what does it mean? Just you can research and see cuboidal, right? So cuboidal epithelium, right? Cuboidal epithelium. So it is that if I draw it cuboidal, so it's just like cuboidal epithelium. So it's like a, right? So if I draw, it is, this is the better diagram, cuboidal epithelium. So I'll erase others, right? Cuboidal epithelium, like this, right? Like a cube, right? So covered by layer of cuboidal cells, germinal epithelium, right? So now here over is divided into cortex and medulla, cortex and medulla. So now here, so that you know that this is a 6 milliliter structure. That means extremely small. So 3.5 into 2 into 1. Very small, you can see. So in here, that means you get everything. Right? So that you can see now here in this diagram, the cortex, the dark part, cortex, and the medulla, so mid part, right? So you can see here oocyte last week we discussed, so that you get somewhere tunica albuginea, right? The important things, this definition. So where's the tunica albuginea and the function of it you have to know, right? So ovarian ligament, functions of the ovarian ligament. So medulla, corpus luteum, developing corpus luteum, corona radiata, so theca folliculi, ovulatal oocyte, zona pellucida. So that is, this is oocyte, anthem, vesicular Graafian follicle, right? Mesovarium and blood vessels. So you get a, granulosa cells, late secondary follicle, so that you have to pronounce it properly. Sometimes in Viva they can ask, but the important thing is those are not bigger as this. You can see, as I said, it is 6 centimeter. That means a 6 centimeter cube volume. So it's a very small, but you can see, right? Right. So the volume of ovary, right? So now here ovary cortex, primordial follicle. So I said that now here there's a cortex and medulla, two parts, right? Primordial follicle, so you get oocyte, so contain an immature ovum with large vesicular nucleus and prominent nucleolus. Primary oocyte covered by single layer of flattened follicular cells. This is primordial follicle, so follicular cell, nucleus, oocyte. So this is it's a this diagram is a real diagram, colored real diagram. This is the drawing, right? So this one, primary follicle. So you know that I said the cuboidal is like a cube, right? Oocyte, increase in size surrounded by zona pellucida. Secondary, right? Secondary follicle. Follicular cells form multiple layers, right? Multiple layers. So granulosa cells. So you have to know about these things. Primary follicle, zona pellucida. So follicular cells, basement membrane. Then follicular cell form membrane granulosa, oocyte, zona pellucida. So this is called secondary follicle. Then you get preantral follicle, zona pellucida and follicles, right? Next one, tertiary follicle. So then you get the follicular cavity, right? So you should be, right? So those things you need to write it down. Primary follicle. That means you should be able to describe it. But those are not enough what is in the diagram. So then you have to get secondary follicle. What is the difference between primary and secondary follicle? Then preantral follicle, right? and tertiary follicle. So how to compare these four structures, right? So those things you need to know, right? So we'll discuss those things later. Ovary, mature follicle. So we call it the Graphian follicle. So normally in the exam, so they give these two terms. So mature follicle or within bracket Graphian follicle. Or just they will say mature follicle, right? It is the largest follicle, right? So now you have to know about these terms. So I'm going to explain these terms later once I discuss that, right? Cumulus operus, then corona radiata, theca interna, theca externa. So what are these? So those are the, so you can see these diagrams. So those are the what you see, right? So mature Graphian follicle, theca externa, theca interna. So externa is the outer layer, no? So then anthem, granular cell, corona radiata, and the cumulus ooporus. Ooporus. So here that is the stalk which connect the granulosa cell, right? So these function of these things to histology, that means a structure of the cells and the function of those things you have to know. So this is the actual one. So actually that means that they have done very well. So this is the diagram, right? Next one. Ovulation. So we have heard about ovulation. So ovulation I told, so that mammals, so they have ovulation, right? So now here shedding of the ovum from the ovary. So time of ovulation 14 days before the onset of next menstrual cycle. That means so before menstrual 14 days before periods. So you release the ovum, right? You release the ovum, right? Right, hold on. Right. So next one. Stage of meiosis at ovulation. Completion of meiosis 1. So you need to remember meiosis 1, resulting in secondary oocyte and first polar body. Mature follicle ruptures and release secondary oocyte. Those are very important questions, which is stop at metaphase 2, right? So you can watch the previous videos to understand those things, right? Okay. Next one, ovary again. So that ovary, so that you know medulla, right? We discussed that corpus luteum, ovarian stroma, right? You can research this all from the textbooks. You can read it and understand. Next one, corpus luteum. After ovulation, the follicle collapse and transformed into corpus luteum, right? Follicle collapse, right? Corpus luteum of menstruation, corpus luteum of pregnancy, right? So these two things, so you have to know the difference. Corpus luteum of menstruation, corpus luteum of pregnancy, right? Next one. So this is the hormonal, regulation of the ovulation. So hypothalamus impulse, the pituitary gland, gonadotropin. So you get FSH and LH. You can see now here, FSH, LH, both, they work on maturation of the follicle, ovulation, and corpus luteum. So now you need to understand, right? So if you have, defeating, secreting, that means if you have some problem in the pituitary gland or hypothalamic impulses, if you have some problem, so what will happen? So if you have less FSH and less LH, there are conditions you will meet those things in gyn and obs, so sometimes you learn in pediatric clinics and endocrinology clinics, so those things. Various illnesses happen if you don't have enough FSH and LH. So over secretion or under secretion. So these are normally managed by the endocrinologist. Right. Next one. So now we need to talk about fertilization. Right. So fertilize, so this is the ovary. I told it's a very small. These are very small, right? Ovary. So then here you ovulation. So then unfertilized ovum only you have to know. So that here you get a fimbria. So that remember that they can ask function of the fimbria, function of the round ligament, function of the ovarian ligament. So those things. So quickly note it down. So you have to additionally study those things. Right. So then here you get the fertilization. So then you get a zygote, then first meiotic division, then you get the second cell stage, So that means about 1 day, 1.5 days. So 16 cell morula, that means 3 stage, 58 cell blastocyst, and 107 cell blastocyst, so 4.5 days. Right. 6 days partially implanted, so early bilaminar blastocyst, 6 days. So you get here uterine gland and uterine mucus. This is the uterine wall. Right. Process of fusion of two mature germ cells and ovum and spermatozoa form a single cell zygote. Usually ampulla or lateral 1/3 of uterine tube. So somewhere here, no, lateral 1/3 is here, right? Only one sperm, human. So if you had listened last slide, so one sperm pierce the, only one sperm fertilized the egg. Only one sperm pierce the zona pellucida, and it has acrosome. Even you have learned this from your O levels. You have acrosome. So acrosome has enzymes. So acrosome, there's a cap also. So acrosome release those enzymes. So then it dissolve the zona pellucida and enter the ovum. So after entering the ovum, only second meiotic division take place. It initiates meiotic division of zygote, resulting in the formation of the multicellular organism. That means us, right? So stages of fertilization. So stage of fertilization. So if you get sick of your life, you need to if you get sick of your life, you need to think how you have survived. From the millions of sperms, right? Most strongest one has had the ability to fertilize. That means millions of your friends have died on the way. So it's not a 1 kilometer, 2 kilometer thing, no, few centimeters from here to here. Because sperm is alkaline, so vagina is acidic. So there's an immune system, and there are so many barriers that which prevent the sperm. Because why that it has so many obstacles? Nature won't strongest sperm to be fertilized by the ovum. But the thing is, the funny thing is what? In ovum or egg, right? Egg, human egg doesn't, that means we don't have a choice. See, it's simply you can think like the male doesn't have a choice. For one egg, right? Millions of sperm. Whether it is good or bad, one will fertilize. So that means, so ovum has a choice, but sperm doesn't have a choice. It is how the nature has, because you can relate these things to your day-to-day life as well, because it has all same pattern. It has all same pattern. No choice for the one egg. Millions of sperm fight. So it just simple as for the one girl, several boys try on. So it's the same thing, right? So now here, so only the straw. That means if you get sick of life, you think like that. Millions of other competitors have died, and one of your father's strongest sperm penetrator. Once it fertilizes normally, right? It's very rarely only 2 or 3 sperms goes. So then something happened. This embryo does not grow or die, or there's other issues we'll discuss about abnormalities in fertilization. But normally one sperm fertilize the one egg, right? So this is the stages of fertilization. So this is we have learned it for our A level as well. Sperm make contact with the egg. Acrosome react with the zona pellucida. So hydrolytic enzyme, you get protease, so those things, right? So acrosome react with the perivitelline space. So here that you get is the space, right? Perivitelline space. Plasma membrane, sperm and egg fuse. Sperm nucleus enter the egg. Cortical granules fuse with egg plasma membrane, which renders the vitelline layer impenetrable to sperm. Right. Stages of fertilization. Right. So you have no capacitation, final step of sperm maturation. Acrosome reaction. So release acrosomal enzymes that facilitate penetration of the zona pellucida by the sperm. Right. So now again the steps are important. So you have no step 1, sperm binds the zona pellucida, acrosomal vesicles, content of the acrosome. Release exocytosis, acrosomal reaction, right? So then acrosomal content. So here then hydrolytic enzyme release help the sperm burrow through zona pellucida and enable binding and fusion of the sperm with the egg. The sperm nucleus enter the egg cytoplasm, right? So that's how it goes, right? Barriers of sperm. Patent of the corona radiata, patent of the zona pellucida, fusion of the oocyte and sperm cell membrane. You know, this is very, very difficult. Some sperms die here, some die here. So only the strongest one who had proper enzyme, proper amount of enzyme, who had proper energy, right? So because you have to penetrate those all barriers and you have to penetrate the corona radiata, you have to penetrate the zona pellucida, right? So it is not something, so you can see that corona radiata is the outer membrane. So it's a, you can see projections. After that you get zona pellucida. So that lot of sperms stop from here. Only one can go in. Right. Fusion of the gametes. How the cytoplasm of the mature ovum contain two pronuclei. Their envelopes disappear and their chromosomes arrange themselves in the equatorial plane. Right. So nucleus of the ovum, so corona radiata, zona pellucida, first and second polar bodies fertilizes sperm. Mixing the cell nuclei and the chromosome of ovum and sperm, then fertilization is complete. So fertilization, so we'll discuss more about that. Now here we'll discuss additional information. So results of fertilization, completion of second meiotic division of the female gametes. Restoration of diploid number. So you know that human has 46. So father's sperm has 23 and mother's egg has 23. So all 46 of chromosome. In the zygote, determination of the chromosome sex, initiation of the cleavage of the zygote. So that what it had been colored, it's better you to know, right? In vitro fertilization, because sometimes what happen, so there are various conditions. Sometimes motility of the sperm is insufficient, and also sometimes there's there are issues. Sometimes outer layers of the egg is too thick. Another thing is acidity of the uterus is too much. So in those conditions, you can't have a baby at normal way. So therefore, what happens? You take that means you stimulate. So I'll explain those things later. So you stimulate the ovary. There's a medication for that. You get a hormone. Then you get the ovum. You retrieve from those. There's a small surgery. You take the ovum. And you take the sperm from the father and you fertilize by the embryologist. So this is the process of the fertilization because you actually you select the good egg and the good sperm. So you look through the microscope and you select them. And then sometimes then you can add enzymes also that to make sperm that means fertilization easy because that if you have thick membrane you have to make it thin. So you can add those enzymes now you can buy it. Right. So buy means we can't buy, but embryologists, the labs they have. So then you can thin this membrane and you artificially you fertilize. So then normally you fertilize few eggs, about 3, 4 sometimes. So you keep, those days you keep 3, 4 eggs, fertilized egg in mama's uterus. But there's a chance you get 4 babies because because sometimes you kept 4 eggs but only one grow. But sometimes what happened you get 3 babies. Now I heard I don't know exactly. I'll see now. Now they say about 2 fertilized egg you keep because if you get multiple pregnancies it's difficult for the mother. Right. So this is in vitro. Now therefore that if your sperm is not well developed or not well motile, and under those conditions, if you have any blockage in fallopian tubes, if there are some problem in the fertilization, so that you can do this. So this is also successful to some extent, not 100%, but it's successful method. It's very expensive, but now you don't have to worry about if you can't conceive in normal way. So now there's a in vitro fertilization, right? Fate of the zygote. You get a 2-cell stage, 4-cell stage at morula. Right. After fertilization, the zygote undergoes successive processes of cleavage division, formation of the morula and blastocyst, and subsequent implantation in the uterine endometrium. These occur during the first week of development. Right. Cleavage. So you need to know about the meaning. So all topic you have to write in piece of paper and you have to research and write in piece of paper and you need to study additionally. Process of repeated mitotic division of the zygote within the zona pellucida. Now, if they ask you to, because sometimes there's a specimen, you have to go through the specimen and in test. So they will ask you to identify and write in piece of paper. This results in a number of small cells known as blastomeres. So this is 8 cell stage, 16 cell stage blastocyst. Right. Cleavage. So these things ours, this is important if you are trying to become an medical embryologist. So 2 cell state 30 hours of fertilizer, 4 cell state 4 to 50 hours, 8 cell state 72 hours. So 16 cell 96 hours. So these are these specimens normally in the in laboratories you will find this specimen fixated specimen. Right. Next one, morula. Morula is about 16 cell state. The mass is called morula, where all the cells are apparently similar in size and appearance. Zygote migrate from the ampulla. Zygote migrate from the ampulla, right? Ampulla and reach the uterine cavity during morulation. This occurs approximately 72 hours after fertilization. Means 72 hours after fertilization means what is it? That means about 3 days. So there's a term, right? Ampulla. So that means if you go back, you can find ampulla, right? Okay, you can research and find about ampulla. I don't know whether I can find it here. Ampulla, right? Here, no? Ampulla. So ampulla, isthmus, and intramural, right? Those things you have to know in fundibulum, right? Those terms you have to say repeatedly, then you remember, right? So now here, morula, this occurs approximately 72 hours after fertilization, right? Blastocyst. Right. Okay. Now you can see the significant difference. Cells arranged as inner and outer cell mass. So inner cell mass. So it is an embryoblast. Forms embryo proper. So other one is the outer cell mass. So trophoblast. Forms the protective and nutritive covering of embryo. It collects between the inner and outer cell mass to form a fluid-filled cavity blastocyst. Blastocyst formation between 4th, 5th day after fertilization. So you have to know what is blastocyst and what is inner cell mass. That means embryoblast, right? Embryoblast, and you have to know trophoblast, right? So you can see now here trophoblast is here, then embryoblast is other side, right? Right. So hatching of blastocyst. Thinning of zona pellucida in a day 4 and disappearing on the day 5 after fertilization. Initiate attachment of uterine epithelium 6th, 7th day of implantation. Site of implantation at the junction of fundus and posterior wall of the body of the uterus. Uterine endometrium is now called, that is the other important thing after implantation. So decidua. Trophoblast is implanted in a decidual wall by proteolytic action, right? So you will see this later, right? Next one, abnormal implantation, right? Abnormal implantation. There's a condition, please write it down somewhere. Intrauterine placenta previa. Extrauterine ectopic pregnancy, tubal pregnancy, you get ovarian pregnancy and abdominal pregnancy. So you will see this. This is the normal placenta. You can see where it should be. Then you get other one, low lying placenta, right? Less than 2 centimeters from the cervix. So it is somewhere here. You can see now here the direction. This is a little bit, it has gone down. No? Placenta previa. Placenta completely covering the cervix. That is some abnormal condition. This is the all abnormal, but this is serious. But now, okay, see, normal placenta, placenta previa. Right. So write it in a piece of paper. You can see this here in the Cleveland Clinic. So you can download these images from there, right? This is the placenta previa, right? Twin pregnancy. Twin pregnancy, there's a, two ways. So it had been formed. Two eggs fertilized by the two sperms, right? So this one, you can see there's a significant difference, right? Dizygotic twins, so non-identical twins. Non-identical twins. So you can see now here, if the two eggs fuse with the two sperms, so those are non-identical. See, right? This is non-identical. This is identical. That means after, right, two cell stage, what happened? So in simple, if I say, so what happened? So it break into two. Right, break into two, but it's not good to use the term break. But remember now this, so there is no rule that, so this is the final result. This is the final results, because from the zygote you don't get like that here, because from the two cell stage you don't get like a human form like that, but this is the outcome, right? Because this is A and this is B, right? So don't link, this is the continuous diagram, right? It is not like that, you can see, so that they have separate A, B, C, D, right? So this is right. So now here, this one. So two zygotes have totally different genetic constituent. So this is two zygotes, right? Two zygotes, right? So twins have, these are called dizygotic twins. A, dizygotic twins. A, right? So zygote implant individually and the uterus usually each envelope is own placenta. So you have your own placenta. Remember, the placenta and umbilical cord is different. Huh? The small one here, that like a tube one, this one, umbilical cord, this is the placenta. Okay, right? Now here, write it down, umbilical placenta. Those are two things. Huh? Right. So monozygotic twins are identical. Now here, zygote divided into two. Then they share, splitting the zygote at various stage of development. Right? So this is monozygotic. They are genetically almost equal. That means easily you can transplant. And also it had been found, even this, monozygotic twins, they grew up separately. So still they share common things. So there's a report, I don't know whether those are correct or wrong, that what I read in a report, where one twin got a heart attack who grew apart, far apart, and the other also got a heart attack at the same period. So it may be possible, right? So this is the separation placenta. So this diagram, so we can two cell stage zygote. So this is how it is developed, right? So you can see now here. So this is okay, right? This is separate placenta and chorion, right? Fusion of placenta and chorion. And this one you can see, so two separate babies, right? Two separate babies. This also two separate babies, obviously. But okay, so now here, let's see. Now you can WhatsApp me. So which diagram, or you can send me a message to the Zoom, right? Which is the monozygotic twin and the dizygotic twin. So if you look at, you will now realize, right? Monozygotic and dizygotic. So you can see now here, right? So what is it? Right? Okay, later on we'll discuss about these things more. Monozygotic, dizygotic. So that if you look at this diagram, so you will understand. So it's monozygotic and the dizygotic, right? Right. So here this diagram, if you realize, you can see now here, this is the separate placenta and chorion, here fused chorion, right? So here you get the common placenta, So common amniotic cavity, right? So if you look this, so you will, so that when you are studying the diagram, it is better. So you study like that. You divide and study like that. Then it is, so then you get right? So then you get the easy picture. Because if you study everything separately, then that is going to be a problem, right? So always when you do, you relate it to the previous diagram, right? Conjoined twins, right? So I'm going to show you some conjoined twins survive. I'll tell one of my example, known example, Iran. So there are conjoined twins. They lived up to, I think, up to 30 or 20 years old. And during the surgery, I think it is craniopages. Craniopagus Iran. So we'll watch that video now. So craniopagus, that means in simplified, say the connected, there are two heads, skulls are connected. Okay, we'll watch that. It's a very right. Some people, even certain, thoracopagus and pygopagus or pigopagus. So, depend on the severity, sometimes they can separate conjoined twins, right? Now we'll watch about craniopagus twins, right?
More confident. No, just more confident. Sorry, how that feels. Okay, let me introduce you to the twins. Let me introduce you to the twins, okay? The young lady on your left, okay, is...
Lale and Ladan woke up quite early, at about 7 o'clock. They had a shower, they prayed with some of their good friends, and they were in a very upbeat mood. A lot of the hospital staff visited them to give them their best wishes.
And then they will shift, 你看到了how long the shift will be.
No, yeah. Medical team is going to discuss and then I think this is the day that they have been waiting for, obviously. So I think they're very happy that the day has finally come. I mean, obviously, I think they would probably still be quite nervous, right? Anxious. They probably have a lot of thoughts going through their minds, right? But yes, this is the day they've been waiting for, and I think they are coping very well.
Right. So now you know, right? Now you know that that's a real story in Iran. They died actually during the surgery. They died. So that is craniopagus, right? So pigopagus means so from the back it has been connected. So here thoracopagus, that means from the in this area it had been connected. So you can research more about thoracopagus, right? So now here, next one. See, normally they don't survive as you can see. So they don't survive, but some survive too. But even if they survive, it is the normal, it is death, right? So now here, that is the end of the session, right? So now here we'll continue, we'll revise it a little bit more, right? So stage of fertilization, and so those things you need to reread, right? So then only you get more information about it. That means by just looking at it, looking at the slide may not help you. You have to research. That means infundibulum, where it is location. So function of the fimbria, round ligament, ovarian ligament, intramural, isthmus. So that sometimes you have to know about the histology of that also, how the cell has been, the tissues have been arranged. So those things, right? Okay, now